at a time. Now, we're going to follow 2 genes at a time. So this would be a 2-factor cross, right? Following inheritance of 2 different traits in an individual. Now, Mendel crossed pea plants, all right? And remember, those pea plants can only have 2 different traits, right? Flowers could either be purple or white. Pea pods could either be green or yellow. So when you cross them together, right, he would expect a particular pattern to emerge. There's actually 2 different patterns we can start seeing emerging when we have a 2-factor cross, right? Mendel typically saw that when 2 genes are crossed together, those genes were independent, right? They were randomly distributed into gametes. But as we'll see in the next few slides, researchers that expanded upon his idea also saw a different pattern that instead of the genes being independent, they were linked together, meaning they would always be passed down as a unit, as a pair, right? So these 2 different methods of being passed down. Right? So if we see genes that were not linked, right, that were independent of each other, typically those genes are going to be found on different chromosomes or on one chromosome but far apart from each other. Versus if those genes were linked, typically those genes would be on the same chromosome and they would be very close together. Right? And we'll keep expanding upon this idea here. So if we are following 2 different genes, or if we expanded this to 3 or 4 or 5 different genes, right, we could start to see patterns that they're either independently assorting, right, follow the principles of independent assortment, they randomly line up on the metaphase plate, right, or that those genes are linked and they're always inherited as a unit. All right, so if we look just a little bit closer at linked genes, and we'll get into this more in a few slides, right? But genes that are linked, they have this physical association with each other, right? They are very close together on the same chromosome. They are always going to be inherited as a pair, right? When crossing over occurs, they typically don't break apart. They'll go together as a unit, even in crossing over. Right? So this is found linkage, right? When 2 or more genes are closely associated with each other and are always passed down as a unit. Now, like I said, typically a linked gene is going to be on the same chromosome and they're going to be really close together. If we have genes that are on different chromosomes, right, they're not going to be linked. If they are genes that are far apart on the same chromosome, they're also not going to be linked. They can only be linked if they are found together on the same chromosome, found very close together on the same chromosome. All right, so we have these 2 different patterns that we can see when we follow 2 genes. They're either linked or they're independently assorted. If we take a quick look at what this would look like, right, if we followed through linked genes, through meiosis, and got an F2 generation, right, we would see that the parents here, right, these are true breeding pairs, meaning they are homozygous for 2 traits, right? They are passing down their alleles to that F1 generation. But now, instead of this F1 generation having 4 different possibilities of ways that alleles can be combined, there's only 2 possibilities. Because the dominant alleles, Y and R, they're linked together in one parent. The recessive alleles, they're also linked together on the other parent. Right? So they can only have 2 possibilities when they produce their gametes. Right, we're going to, again, expand upon this a little bit more. Right, so they're homologous pair, right, from mom and from dad. Capitals are on one chromosome, lowercase are on another chromosome. We can't break them apart and redistribute them because they are linked. They're always going to be inherited together. So capitals are always inherited together. The lowercase letters always inherited together. which is very different than what we have been talking about with a one-factor cross. Now this is giving you an example of a two-factor cross. Right? Again, the parents, they are homozygous for their traits. Right? They are making that F1 generation that is heterozygous. When we cross the F1 generation with itself, right, we see we have many more possibilities of gametes that could be produced and thus resulting offspring. Right? Many more possibilities because they're not linked. Right? So either the genes on these individuals are far apart on the same chromosome, or they're on totally different chromosomes themselves. Right? Big picture here, many more different combinations are available because they are not inherited together. And again, we're going to come back to this. This is kind of my quick introduction to the concept. All right, we're going to start by looking at independent assortment, right? Because that's what we've been talking about. That's what's easy to talk about, right? So we know that linked patterns can occur, but we're going to focus on the independent assortment pattern first. When we do a two-factor cross, right, just like when we did a one-factor cross, we get a certain ratio that we would expect, all right? So this time we're following two genes, right? We would expect a ratio of offspring in a 9:3 to 3:1 ratio, right? So 9:3 to 3:1. And this is for the F2 generation. Now remember, the one-factor cross, we would expect a 3:1 ratio in phenotypes and a 1:2 to 1 ratio in genotypes, right? That was expected. The 9:3 to 3:1 ratio is expected for the two-factor cross, right? Whenever we see this ratio occurring in offspring being produced, we know that this is consistent with the theory of independent assortment. So again, they're randomly lining up on that metaphase plate in meiosis. This ratio will become important in a few slides when we start talking about linked genes. Right? But I want to point out, just like for the first factor, the one-factor cross, two-factor cross also has this very expected outcome. Let's look at how to do a two-factor cross as a Punnett square. Right? So now we're adding in some more complexity. So we're going to do a cross between two F1 generation individuals. So in the F1 generation, we're crossing two individuals with the genotype capital R, lowercase r, capital Y, lowercase y. So the very first thing we have to do is figure out all of the possible combination of gametes that could be passed down. Now remember, we're going from a individual here that's producing gametes, right, producing 4 daughter cells, right? So, and because we have 2 different genes we're following, this means there are 4 different unique combinations that can occur, right? One of those combinations in the gametes could be a capital R and a capital Y, right? We have to keep one allele from each of the genes that is going to be passed down. We're not just going to pass down the R's. We're not just going to pass down the Y's. It's one from each gene. So capital R, capital Y is one possibility. Capital R, lowercase y is another possibility. Likewise, lowercase r and capital Y is a possibility, as well as lowercase y and lowercase r, right? So there's all these different possible combinations, right? Because again, they're randomly assorting on the metaphase plate. We can get a whole host of combinations of our genes. Right? In this case, we only have 2 genes we're following. We can only have 4 different possible combinations. So now that we know the possible gametes that could be passed down, we have to put it along the side. Right? Very similar to how we did the one factor, except now each row and column has 2 genes associated with it, the R and the Y. Right? So for each possible combination, we would put that in each column and each row. Right? And then again, it's just a matter of matching them together. So this first one here, we have a capital R, capital Y, matched with a capital R, capital Y. right? So the genotype that this will lead to is capital R, capital R, capital Y, right? It's just that matching game. Now, a couple of rules will apply when we have two different genes, right? So we're always going to put these same genes next to each other. So it's always going to be RR, YY. It's never going to be RY, RY, right? So we're putting the same, the alleles for the same gene together. And we're always going to write, for a particular set of alleles, the dominant one is going to come first. All right, so for example, here I have a lowercase r and I have a capital R, right? I would write the capital R first and then the lowercase r. We're always going to put the dominant one first. This is just the standard convention of how to do these types of Punnett squares. All right, are we ready for some practice? No. Well, practice is going to be helpful. All right, before we leave this side, again, I could count up the phenotypes, right, and put them into a ratio. I see that there are 9 yellow round, 3 green round, 3 yellow wrinkled, 1 green wrinkled. It's a 9-to-3 to 3-to-1 ratio. That immediately tells me they are independently assorting. All right, let's do some practice. So I want you to cross these two butterflies, right? One is red with a striped body. One is yellow with a dotted body, right? I put their genotypes here, right? The first thing you have to do is for each individual, determine the possible combination of alleles that they could pass down to the next generation, right? So start there, then you'll put them on your Punnett square and match letters together. Give me about 5 minutes, because I expect this to take a little bit longer. What was that? It'd still be 4 rows. But yeah, you're right in that it's the same. Yeah.
I don't get it. You don't even know how to do it. You're watching. You're not even good. We get like really done and then explanation how to do this.
Yes, we are going to go over this together. Yeah, adding in that second gene really throws everything for a loop here. Not necessarily, but you could, yeah.
Sorry that I'm smart.
See, I would think Friday at the end of the week, everyone's excited for the week.
I just think God.
Yeah, well,
Thanksgiving break is just around the corner.
You guys wake up to fire alarms 4 times. I didn't wake up a single time.
There's
Oh,
Yeah.
I gotta forget.
There was one
I
at
only
like
did
12:30
Oh.
ish, and then one at 1, and then one at 1:30, and then another one at 2 ish.
What was causing it, you know?
People were
Yeah,
playing.
you guys know.
Yeah, no, that if counting those,
Who said that?
set 8 or 9 fire alarms pulled this semester.
Wow.
They're not very good at avoiding it.
Is that you?
Yeah, no,
I hope they know who pulled it and
No,
there's
they
some
don't.
ramifications.
You need to get the fire alarms with the UV stuff. When you pull it, it stays on your hands,
Oh.
so they can just scan everybody's hands when they walk in, and then they know. Because it's a class D felony.
Yeah.
You can get in big trouble for pulling it.
Yeah, I skipped my 8 a.m. this morning. I emailed my professor like, yeah, sorry, I'm not.
Don't do that. None of that.
I would like some extra sleep.
When I was a freshman, I was in sorority, and they were playing some sort of like kickball in the hallway on one of the
Yeah.
levels, and they hit the sprinkler and flooded like half the dorms on the right side. That caused chaos too.
Yeah.
I didn't have too many parties.
At least somebody wasn't doing that on purpose.
No, but I think there might have
There's
been
also
other
people in
influences.
5. There's also people in 5E. Like I think yesterday or something, all of 5E got written up in co-ed because they were all being loud.
You're not talking. You can't shop GLP.
Wednesday night, campus safety had to walk through my room because they were smelling smoke on our floors.
Before
Appeal for the campus safety officers. They have a
Or
group.
what class?
Well,
Okay.
All right, let's
Before
go
E.
through this together.
It was
So,
just her.
first thing we need to do is determine the possible allele combinations that can be passed down. Let's start with the hard one. Let's start red butterfly, striped body. All right. They each have to have a R allele and an S allele. Right? What is one possible combination that we could make?
Big R, little.
Big-
No.
Not big R, little r.
Big
R,
What are you talking about?
So we'll start with big R, little s. Right? That's one possibility. So we could match these two together.
We mentioned the same one.
All right. So big R, little s. Maybe I'll do a different color here. All right. What's another possibility for an
Big R, big
allele?
S.
Say that again.
Big R, big S.
Big R, big S.
Oh,
Yes.
look at me.
All right. So we can match those two together. So big R, big S.
John Wick style.
All right. What about we haven't used the little r yet. What's
Little
another
R, big
possible?
S.
Little R, big S.
Little r, big S, little r, little s. Yes, those are other two possible combinations. All right, so we would write those out as well. All right, now we have our possible combinations. We could write these combinations, one for each column in our Punnett square. Right, what about the possible combinations of alleles for the yellow butterfly?
Little r, little s.
Little r, little s. That's the only possible combination.
Wait, that's
Right?
the
Again, we could go through and say this r is pairing with this s, this r is pairing with this s. Same for the bottom, but it's the same thing. Yes, it leads to the same genotypes. All right. So now again, we could write that on the opposite side here. There we go. So we would write out our different allele combinations along the top for the red butterfly, along the side for the yellow butterfly, and then again, a matter of matching them together. So this first match here, right, we would have big R, little r. I put the uppercase letter first because it's the dominant allele, but I keep those R's together. Same with the S's, big S, little s. All right, if we look at our next matching, again, we have a big R and a little r. So I'm going to write big R, little r. But I have two small s's. So I'd write little s, little s. Right? I would keep going through and matching them up, just kind of matching the letters together, putting them in the right order, until I have my whole Punnett square filled out. So the hardest part is just making sure we know how to find the different allele combinations. Now again, allele combinations, we're looking at the genotype here, right? But that can be translated into phenotype, right? So if I were to put the pictures over it here, right, that would be the phenotypes that it translates into. Now, you might be noticing, hold on, I thought we talked about a 9:3 to 3:1 ratio. Now, I don't see that here. Well, yeah, that's true. I don't see a 9:3 to 3:1 ratio. I see, actually, the way it plays out is each column has the same genotype. So it's like a 1:1 to 1:1 ratio, right? That's fine. I'm not concerned about ratios here because I am not looking at an F2 generation. This is just a random cross. I would only expect that ratio to occur when we have true breeding pairs that go down to the F1 generation and then the F2 generation. All right? So yeah, it's not a 9:3 to 3:1 ratio, but that's not telling us anything because this is not part of a true breeding pair generation line. Any questions about this two-factor cross? All right, let's talk about when our genes do not follow the rules. All right, so independent assortment, principle of segregation, right? That's what we have talked about so far. But I also told you that the other possible pattern could be that our genes are linked. So traits that do not follow those ratios observed by Mendel tend to be linked genes, right? So we're going to look at an experiment here. An experiment done by Basin and Punnett, the same Punnett that the Punnett squares are named after. They crossed two true breeding lines of sweet peas, right? Sweet peas versus the garden pea that Mendel was looking at. And so they had their true breeding pairs, right? They're homozygous for their alleles, right? For purple flowers and long pollen, those were the dominant alleles. For red flowers and round pollen, those were the recessive alleles. They did the cross. They got the expected F1 generation, right? All of the dominant traits showed up. Their genotypes were all heterozygous. They then crossed or self-pollinated the F1 generation together. And then they got the result, which it's every single possible combination of alleles, but unexpectedly they got a different ratio. They did not see a 9:3 to 3:1 ratio. They saw something else, right? They saw a roughly 15:1 to 4 ratio, right? That's different. That's not the expected ratio, right? So what they're seeing is that the genes were linked, right? What they observed is different than what was expected, and it was different because these genes were linked together. They were being passed down as a unit rather than independently assorting, right? So they are no longer following that rule of independent assortment. When they're lining up at the metaphase plate, right, it's not completely random because the alleles are linked together. All right, so we're not following that ratio. Let's put this into practice. All right, I have given you two lines of crosses here. All right, I want you to tell me which one is linked assortment, which one is following the rules of independent assortment. As a quick caveat here, this cat is supposed to be orange. It has orange highlights, but it did not show up very well. All right, so I want you to, I'll only give you about a minute here because it should be relatively fast to figure this out. Every time I walk past these boxes of brownies, I don't know why they're there, but I haven't had lunch yet. It's making me hungry. I don't see any meeting that happened in here today. Who knows? All right, if we were to look at these two different lines of crosses here, we have the one highlighted in green, the one highlighted in blue. Which one is independently assorting? Yeah, the one in green, yeah. If we were to look at the ratios, which is all we really needed to look at, I didn't need to give you the entire cross, right? All you had to do is look at the ratio. If you simplify this ratio, they're all divisible by 5, right? I would get a 9 to 3 to 3 to 1 ratio. That means they're independently assorted. Versus this other one here, right? If I were to simplify this ratio, I would get a 1 to 7 to 7 to 1 ratio. Different than the 9 to 3 to 3 to 1. What can I assume about this cross? Yeah, they're linked, right? It's all based on those ratios. They are linked. Those genes for hair color and hair length are linked together. All right, any questions? All right, so let's continue on with linkage here. So many traits are linked to sex chromosomes, right? We've already talked about sex chromosomes. We know in humans that's the XX or an XY for male or female here. Sex chromosomes different than the autosomes, which are all the other chromosomes in our bodies. I just wanted to quickly point out, because I think this is not a well-known thing, that the XY system that we use in humans is not used across all species, right? So, for example, insects, grasshoppers here, they use an XO system. So females are XX, males are just X or XO, right? Many birds, they use a ZW system, right? So males are ZZ, females are ZW, all right? There's different ways that we can denote the sex chromosomes in different species. I think it's interesting, kind of a fun like trivia question here. If we look at our sex chromosomes, and we're going to focus on mammals here, right, and in humans, the X chromosome is larger. It's larger and it carries more genes than the Y chromosome. So what this means is that many genes that are linked to a sex chromosome are X-linked. Simply because it's a bigger chromosome, it carries more genes. There are traits that are linked to the Y chromosome, right? There's just fewer of them because it's a smaller chromosome, right? Now, we've talked about individuals being homozygous, so having the same copy of the allele. Individuals being heterozygous, having 2 different copies of an allele. You could also be hemizygous, right? Meaning you have only 1 copy of a particular gene, but you have 2 different genes per se. All right, so this would be for males. We have an X and we have a Y. Males are hemizygous. Lots of words starting with H here. All right, so again, when we think about how is this sorted out into gametes here, right? If we have our X chromosome and our Y chromosome, they'll go through the process of meiosis, separating out the homologous pair. They are homologous to each other. They both contain information related to your sex. So red one here, blue one here, then they're going to separate out our sister chromatids. So as a result, we have 4 daughter cells. Each have 1 chromosome or a sex chromosome. Either they have 1 X or they have 1 Y. All right. So I mentioned that a lot of traits are X-linked simply because it's a bigger chromosome. We're going to go through an example of how we would look at X-linked diseases, right? Specifically, an X-linked disease that is recessive. So we're going to look at the disease of hemophilia, meaning your blood does not clot normally. So when I write out the genotype, when I'm looking at sex chromosomes, I have to include the X and the Y. So for females, we're always going to have two X's. For males, we're going to have an X and a Y. Then the genes are going to be superscripted above the X and the Y. All right, so normal female, right, does not have hemophilia, right, is homozygous dominant, has two capital H's for the alleles above the X chromosome. A female with hemophilia that is homozygous recessive has two lowercase h's above the X's. We could also have a female that we would say is a carrier of the disease, right? So this is a recessive disease, meaning it won't show up unless both alleles are recessive. Here, we have a female that has a dominant allele and a recessive allele. The dominant allele is masking the recessive, so she doesn't have the disease, but she carries the genetics for the disease. So she's a carrier for that allele. Now males, because it's an X-linked disease here, it's only going to show up on the X, so we're only going to put the gene superscripted above the X. The Y is going to be left alone because it's not Y-linked. All right, so if we were to look at what this looks like in a homologous pair, right, female XX here, right, capital H, lowercase h, one of each type of allele. This female is a carrier. And a male here, this male has hemophilia, only has one H because it only has one X chromosome, right? So they carry the disease and the disease is expressed. So we could do Punnett squares just like we have done for one factor crosses, but with these diseases that are linked to a sex chromosome, right? We simply have to determine what possibilities can be passed along, put them into the proper columns and rows, and match them together, right? But keeping in mind that if it's an X-linked disease, there's not going to be an allele for the Y. So I think best way to do this is to practice. So we're going to practice this with an example using color blindness, right? So color blindness is a sex-linked trait, right? Being colorblind, right? Colorblind is the recessive allele. I'm going to let you determine, is it linked to the X chromosome or the Y chromosome, and then figure out the genotypes and make our Punnett square. So again, I expect this to take a little while, and we will do a thorough review at the end. Can we do it? That one. So remember, we have to put in the XY chromosome. And what does that mean about the male? Does it have two alleles for this trait? No, it's only on the one with the X.
Sally, I'm there.
I don't think I know anybody.
Because in middle school, we did this as a
Actually, no, you're right. I only know. What?
I think it's kind of weird.
I thought they were you.
I have no idea. Oh, I did.
So we're gonna come close to the edge. Close. Hang in there. I have a scratchy piece of paper. Yeah, when you write down each
Buddy, we're not even doing finish. You're supposed to finish. Let's go. I'm not no, on no, every second. At least it'll cheat on tests. Who are you looking at from the test? Who are you looking at from the test? Andrew, who else are you looking at? Yeah.
someone doesn't have it.
Boys, stop
But
playing
in
me
our
I need to
example,
put it
Russ
in.
does have that recessive trait.
Stop.
Oh, you did it wrong.
Yep.
Where'd you get big C, little C from?
All right, so let's go over this. So first of all, we have to determine, is this an X-linked or a Y-linked trait? Right, what did we come up with? It's an X-linked trait, right? The female has this trait expressed. Females only have X chromosomes, right? So it has to be an X-linked trait. So the female, all right, we're writing down the chromosomes. Male would be XY. What would the genotype of the female have to be? It has to be homozygous recessive, yeah. So 2 little b's, right? Because that's the only way a recessive trait can be expressed, right? She would not be able to have a capital B and a lowercase b, because that capital B, the dominant allele, would mask the expression of the recessive trait. The only way the recessive trait can be expressed if it's the only thing in the genotype, right? All right, so the male has normal color vision. What is the genotype for the male? Yeah, capital B. Do we have anything on the Y chromosome? No, because this is an X-linked trait, so it's not on the Y chromosome. We're going to leave this one empty. All right, so now what we have to do is put it into our Punnett square. Okay, I'll put the female on the top row here. Right, and again, I'm not putting anything on the Y chromosome because that gene does not exist on the Y chromosome. And then it's just a matter of matching them up again. So again, I'm going to follow the same convention where if there is a dominant allele present, I'm always going to put that one first. Right, do the same for the male offspring here. All right, so if we look at the possibilities for their offspring, right, would the female offspring have a possibility of being colorblind? No, because they're always going to be heterozygous. That's the only way that their genes can combine together. And because they're heterozygous, they aren't expressing the trait, but they are a carrier. So they have the genes for the trait. It's just not being expressed. Right? What about males? Are they going to have normal color vision?
No.
No, they're going to be colorblind 100% of the time, right? If they produce a male, it's going to be a colorblind male, right? And we often see this in color blindness, right? It is an X-linked trait when males are born, a lot of them have color blindness if it runs in their family, right? Because they only need one allele that's recessive. They don't need both alleles to be recessive like females do, right? So this is a very classic example of an X-linked trait here, right? So if we did it all out, right, we would have something that looks exactly like what I wrote up on the board. Any questions? All right. I would say practice this, right? Find examples online. This is fair game to put something like this on the exam, right? Give you an example, try to have you figure out is it X or Y linked, and then make a Punnett square of the possible resulting offspring. All right. So far, we've been talking about genes that are either going to be expressed or they're not. They're either dominant or they are recessive. Genes are more complicated than that. All right, there's a lot of different complexity that can go on, and we're going to cover just a few of those different cases, right? One of which is incomplete dominance. So if we look at the parents here, right, one flower is white, one flower is red. If we look at the alleles, right, they are both capital letters. They are both the dominant allele, right? So white is the dominant allele, red is the dominant allele, right? So when these individuals are mated together and create the offspring here, we get a blended phenotype, right? White is not dominant, red is not dominant, but rather they're blending together to create a third color, and so they have this pink color to them, right? They each have an incomplete dominance. Now, if we were to breed this out to the second generation here, we would see an occurrence of the white and the red flowers reappearing, but we would still have that pink phenotype in there. Right? Pink phenotype having one red allele, one white allele, both being dominant. Right? That's different from codominance. So codominance, again, we have parents, and in this case I have used cows, right, where the color of their, cows have fur, hair? the coat color of the cow, right, they both are dominant, all right? So again, capital W for white, R for this reddish brown here, all right? So capital R. They're both the dominant allele. But now, if they are bred together, their offspring are going to have occurrences of both white and brown hair. Right? They're not going to blend together. We're not seeing that the offspring has like a light brown coloration to them. Rather, we're seeing patches that are occurring in white, patches that are occurring in brown. They're co-dominant. All right, so incomplete dominance, it's a blending of the phenotypes. Codominance, both phenotypes are being expressed, and we kind of see this like patchiness of expression. All right, so these are two ways that alleles could be expressed beyond the dominant and recessive. Now, another way that alleles can start to get tricky is that it's not always as black and white as one gene, one trait. We can have pleiotropy, meaning one gene influences more than one trait, right? Abraham Lincoln, he had this syndrome known as Marfan syndrome. This is a very classic example of a pleiotropy here, in that he had one gene that coded for this Marfan syndrome, but it was expressed in multiple different traits, right? He was tall. He had disproportionately long limbs and fingers. He had severe heart problems and an abnormally shaped chest, right? That's all from one gene, right? So that would be pleiotropy. One gene, multiple traits. This is a very classic example. I bet almost every genetics textbook talks about Abraham Lincoln and Marfan syndrome here. All right, so we have a single gene, influencing multiple traits. Now we can look at the opposite end of this spectrum. Right, so far, again, we had been talking about a qualitative trait, right? Either or, it's either a purple flower or it's a white flower. There's nothing in between. One gene, one trait. But on the flip side of that, and on the flip side of pleiotropy, is quantitative traits. Meaning now we have, right, multiple genes influencing one trait. Right, so now maybe we have 3 genes influencing one phenotype, or 5 genes influencing one phenotype. Right, some examples are height. We see in this image here, there are students lined up behind rows of their specific height. Right, we have a whole variety of heights. Skin color, weight, these are also examples of these. Right, so multiple genes, right, influencing one trait, and there's a whole range of traits that can be expressed because it's of multiple genes. We take a look at this from a more genetic perspective, so like looking at genotypes. Right, so my next slide here we'll look at genotypes and how that influences one trait. I see a lot of pens moving, so I'm just going to take a brief pause. So pleiotropy, one gene, multiple traits. Quantitative, multiple genes, one trait. Right, another name for a quantitative trait is also polygenic trait. Right? If I were to ask you what is it where you have multiple genes influencing one trait, and you were to tell me quantitative, that would be correct. If you were to tell me polygenic, that would also be correct. Right? Means the same thing. All right, so here we're looking at the color of corn kernels. And the color of corn kernels is actually influenced by 3 different genes. Right? And we can see that in our pure breeding lines here. Right? If the corn is white, it's all homozygous recessive for 3 different genes. If the corn is all red, it's all homozygous dominant for 3 genes. Right? And then depending on how many of those dominant and recessive alleles show up for each of those 3 genes will determine how red or how white the kernels are. So here's actually a 3-factor Punnett square. I would never have you do a 3-factor Punnett square. I believe you could do it, but it's very tedious, and I don't see a point to it. All right, but I think this 3-factor cross really illustrates, depending on how many dominant alleles or how many recessive alleles, right, you can get a whole range of colors in between. But it's all that one trait, right? So those 3 genes influencing that one trait. All right, let's do some practice. Actually, no, just kidding. I have one more slide. All right, the last way that a phenotype can be influenced is actually the environment. Right? Having really nothing to do with the genes itself, but rather the environment surrounding the individual. So we're going to look at an example of environment influencing phenotype in rabbits. Right? So in warm temperatures, right, versus cold temperatures, we can have different methods of pigmentation for this specific breed of rabbit. So if we look at the warm temperature, so above 30 degrees Celsius, right, that actually inactivates the gene. It stops the gene from being expressed. So if it's stopping the gene from being expressed, it's not going through transcription, it's not going through translation, right? It's not making a protein. So the gene product is not made, which means we're going to get non-pigmented white fur, right? The protein that produces pigments was not made, so we're going to get white fur. Now on the other hand, right, if these rabbits were reared in a colder temperature, so less than 20 degrees, that actually activates the gene, right? That says now we need to express this gene. So this gene is going to go through transcription and translation. it is going to make a protein, right? And that protein is making a functional product, which is pigments, right? And specifically for this rabbit, that means the pigments show up along the feet, the tail, ears, and nose, right? So if you think about which part of this is gene regulation and which part is gene expression, right? What is regulating the gene is the environment, right? The temperature, right? And which part is gene expression? Well, that's whether or not the gene is actually being made into a functional protein, right? So whether or not that gene product is made or not. All right, now we're going to do some practice. So I want you to look at these traits and tell me, are they qualitative or quantitative? It shouldn't take too long, so I've given you about a minute here.
One of my rabbits, his nose hurt. He had a brown spot on his nose and it hurt. It's not all of it. It's just like one that had
You threw my pencil. So maybe do it first and then put your backpack while she starts working. Exactly. This is on the board all the time.
All right, skin tone, is that qualitative or quantitative? Yeah, quantitative. It falls around a whole range of skin tones. There we go. What about body size of dogs? Also quantitative. The ABO blood grouping system? Qualitative. Falls into distinct categories. Now I know there's more than just like O. There's also like O negative and O positive. I just kept it simple here. What about presence of a mane? Qualitative. And we could also make the assumption, because a mane only appears on males, that it might be a Y-linked trait, right? All right. So we talked a lot about different organisms, right? But it's hard to study genetics with humans, right? Simply because we can't get true breeding lines, make an F1 generation, make an F2 generation. It's unethical, right? And it's not practical. So instead, when we look at genetics in humans, we rely on pedigrees, which are a record of ancestry. So pedigrees are normally like a flowchart of symbols here. Circle indicates the female, square indicates the male, right? Normally it's going to follow like just one trait that you're interested in, right? And so if the individual expresses that trait you're interested in, the circle or the square would be filled in. If they don't express it, right, it would be empty. If we know that the individual is a carrier for the trait, meaning they're not expressing it, but we know they carry the genetic information for that trait, then it would be partially filled in, right? So typically we can look back at multiple generations You might have to make some assumptions here, but you might be able to find that you can trace, is this X-linked disease? Is it not linked to a sex chromosome? Is it a genetic thing? Is it just randomly occurring in our family? Right, another classic example, again, hemophilia. We talked about this as one of our other examples. But in the royal family of Russia, right, we could track their ancestry to see who passed along this disease for hemophilia. Right, so Queen Victoria here, right, she was a carrier because it's halfway filled in. Right, she didn't express the disease, but she was a carrier. She passed it along to her daughter Alice. Right? Alice, when she had a daughter, Alexandra, passed it along. Alexandra, still a carrier here. But when Alexandra had a son, their son expressed this disease, right? He had hemophilia. So what we see is that Alexandra, she was a carrier. So she had a capital H and a lowercase h, right, on each of her X chromosomes. And what she passed down was the recessive H. Right? So Alexis here only had the lowercase h, only had the recessive gene, so he expressed this trait. Right? So it's really common to look at pedigrees to kind of figure out what is happening. Okay? Not only can it be sex-linked, we can also trace other linked diseases to some sort of an autosomal pattern. Here we see a pedigree of an autosomal dominant trait for Huntington's disease, which is where nerve cells in your brain will start to die and it can cause a whole host of problems, right? But again, we can see that we look at two parents, right? Male, female is a circle. The male is expressing the trait, which is why it's filled in. You can look at their offspring, look at their pairings, look at their offspring, who expresses it, who doesn't, right? I'm actually going to skip this slide because I didn't have my other class do it, right? And instead, I just want to touch base with you on that pedigrees can have problems to them, right? It's not always simple, right? First of all, if a trait runs in a family, I think I bumped my